Alrighty, hi guys. So this video is about the methods of eukaryotic transfection. So before we actually get into the methods themselves, I want to make sure that you understand what this means. So there's two vocabulary words in here that I'll go over, the eukaryotic and the transfection. And I'll explain those, and then we'll actually go into the methods and how they work. In the past with this class, we've talked about how to do gene transformation to get a gene from outside a bacteria to inside a bacteria. Bacteria are back then we introduced this idea of a prokaryote. Bacteria are called prokaryotes. And the definition of a prokaryote is a cell that has no nucleus. And basically the only example of a prokaryote we've found so far is bacteria. All the other cells have a nucleus that they keep their DNA in, but bacteria, they keep their DNA inside the cellular fluid. It's just floating around with everything else. The opposite of a prokaryote is a eukaryote, which is a cell that has a nucleus. And so animal cells, plant cells, fungi, protists, they're basically everything that isn't a bacteria or a virus is a eukaryote. And so that's us and animals and everything else. It turns out that the methods we've learned in week 10 of putting the gene onto a plasmid and then using the cold hot stress treatment to move the plasmid inside a bacteria to get that bacteria to express the gene, those don't work on eukaryotes because eukaryotes don't use plasmids and they have a nucleus. And so you have a second layer of wall to get through in order to get the animal to use it. Plus, animals, because they don't use plasmids, they also don't replicate anything but their own chromosomes, and eukaryotes don't. And so in order to make a change permanent, last longer than the one generation you put the gene in, you have to permanently glue it into their chromosome, because eukaryotes don't use any DNA other than their chromosomes. So we have to make it part of their chromosome. So it's a little trickier. So the methods, the transformation methods that we learned in week 10 don't work on eukaryotes. So this week we're going over a few new methods that we had to come up with in order to achieve the same results with eukaryotes, which are a little more complicated. And we'll get into those here in a sec. The other vocab word in the title was transfection. Transfection is an overarching term. The idea behind transfection is any method that gets a gene from outside a cell to inside a cell. And you might be thinking, wait, but that's what transformation was. Correct. Transformation is a form of transfection. It's the only one we've taught you so far. So transfection is any method of getting a gene from outside a cell to inside a cell. Transformation is one form of transfection that involves putting the gene on a plasmid and then getting the plasmid from outside the bacterial cell to inside the bacterial cell. So transformation is one form of transfection. Transfection is just any way of inserting a gene of interest into a new target organism. And transformation isn't the only way to do that. So we have a bigger term, transfection, which is any method that gets the gene from outside the target cell to inside the target cell. So we'll learn 5 other methods of transfection. In addition to the transformation method that we taught you in week 10, we're going to go over 5 new methods that work on eukaryotes. They're methods of transfection that all have their own names, just like transformation did. But that's what transfection meant. So this video is about methods of eukaryotic transfection, which means methods that we use, scientific methods we can use to get a gene of interest from outside a eukaryotic cell, a cell with a nucleus, to inside that cell. So the 2 main categories that we go over in this class, we kind of ignore fungi and protists. We go over the main ones, which are animals and plants, because that's where the GMO research is right now. And this week you're learning about animals and plants and how we make GMOs out of them. And these are the 5 key methods that we use to make GMOs out of animals and plants. So this just shows the bacteria transformation method. We go in there, but we can't use it on eukaryotes. So here's the list of 5 methods. 2 of them apply to plants, and 3 of them apply to animals. And I'm going to go into detail on each of these 5 and how they work so that you understand it, because we expect you to understand these in a little bit of detail as to how they work, in addition to what GMOs on your list of 6 GMOs were made, are created by each of these 5 methods. So the first one we'll start out talking about is Agrobacterium. As the name says, agro, as in like agriculture, it's plants, and bacterium. Plants present an interesting predicament, because plants actually, in addition to having cell membranes like animals do, the little barrier around the outside of the cell, they have a wall that's made out of protein and sugar and all sorts of things. So it's this big, thick wall around the outside of the cell that's a little difficult to get through. So plants actually rarely get infected with viruses and bacteria, simply because they don't behave like an animal does. There's no bloodstream. You're trying to get inside an armored city. That's very difficult for creatures. Agrobacterium is one of the few ones that is capable of infecting a plant, but it infects it in a really weird way that we take advantage of to create GMO plants. And I'll go into a bit of detail here on how that works. So here's a little background behind Agrobacterium. So the Agrobacterium method is called Agrobacterium because the bacteria species that we use is called Agrobacterium tumefaciens. A mouthful, huh? I would definitely learn that name. That's the name of the bacterial species that we use in the Agrobacterium method. So it's called the Agrobacterium method because the species of bacteria we use is Agrobacterium tumefaciens. Now, the reason why we use this bacteria in specific is because it has some really cool abilities that most bacteria don't have. Let me see here. Okay, so let me show you how that works. My computer's being obnoxious here. Okay, sorry about that. So this is how it works. So these plants, if you can picture a plant here, I'll make like a tree. Go for a little tree. We've got some green leaves here up on top. Okay, that's a beautiful tree. Down here on the ground, so Agrobacterium tumefaciens is naturally found in the dirt. It's a natural bacteria and it's found in the dirt. And it struggles, or it has the same struggles as everything else. These plant cells have an armored wall around it, but as a plant grows, sometimes the cells down here at the base get scratched by rocks or by animals or something, and so it kind of breaks their cell wall a little bit. And the cells can fix that, but it takes them a little bit of time. So if we zoom in, I'll just zoom in on a few cells here on the ground here. Okay, here we go. We'll zoom in on a few cells on the ground. Oh yeah, turned from a square into a pentagon. That's great. Okay. Basically what's happening is we have these plant cells over here on the left. Got plant cells, and plant cells, they've got these very thick cell walls here. But what we've done is this got damaged somehow. So we've got this one got a little damaged here, a little damaged, this one down here too. So this is right about at the dirt line. And this bacteria comes in here and he's super excited about life because he likes infecting plants. He likes getting plants sick. So he comes in here. This is our little Agrobacterium tumefaciens. And just like a lot of bacteria, he has this little plasmid. It's called the TI plasmid. You don't need to know that. But he has got this little plasmid in him. But plasmids don't work on plants. But that's okay. He works around that. What he does is the agrobacterium gets attracted to the plant because it smells that it was damaged. And then it creates this little needle. So it basically forms this little needle and pokes the needle inside the plant cell through one of the little damage holes. That kind of looks like a scary fish. And then it cuts a piece of its plasmid off here. So we've got a piece of the plasmid, and it basically injects it down its little needle and puts it inside the plant cell here. And what's on this piece of the plasmid, this is just a little background, you don't need to know this, but what's on the piece of the plasmid, there's 2 genes on it. One of the genes makes the cell cancerous. So it actually turns out plants can get cancer. It's called a gall, and it forms this big bulge of cells where the cells are just dividing like crazy. So that's one of the things it does, is the bacteria causes the plant to have cancer. Now, why would a bacteria want to cause a plant to have cancer? The answer is in the other gene. The other gene that's on this little segment of the TI plasmid that it injects is a gene to make bacteria food, basically. It makes a molecule that this particular bacteria, Agrobacterium tumefaciens, eats for food. So essentially, this bacteria injects this thing in here and commandeers the cell to make bacteria food for it. And then it's got another gene on here to make this cell divide so that it makes lots of these bacteria food factories. So it's making the cell make bacteria food and then divide to make more bacteria food factories to feed this agrobacterium and all of his kids for ages to come. That's how this bacteria works naturally. We try to commandeer this idea as scientists to trick this agrobacterium into dropping off the genes we want instead of the bacteria food and cancer genes. So what we do is, remember in week 10, we learned about how to put a gene on a plasmid. So you have restriction enzymes, and we cut them out, or we take the little bacteria plasmid here, and it's got these, we'll put them in red, it's got these bad genes on here, the cancer-causing genes. So this is the TI plasmid here. We come in here, we use restriction enzymes, and we cut out the bad gene, and we glue in whatever gene I'm trying to put on the plant, whether it's like the gene that they took from Bacillus thuringiensis to make BT corn, for example. We can take that gene, let's make it yellow. Oh, that's an erasing. We take that gene and we use restriction enzymes and we glue it into the plasmid. Now this plasmid has the gene that we want on there, the BT gene, instead of the cancer-causing genes. And then we transform it into the bacteria. We do the hot, cold, hot thing, stress the bacteria out, take it up. So now this bacteria has our plasmid, but instead of having the cancer-causing genes, it has our BT gene right here. But the bacteria can't tell the difference. It still thinks it's got the cancer-causing gene and the bacteria food gene. And so it's like, ooh, so it goes up to a plant cell that we've taken the cell wall off of. We purposely do that. And then while the cell is trying to rebuild, the bacteria can come in here and inject, or it forms a needle and it cuts out our little piece here and injects our little piece in here, expecting to have this plant cell now have cancer and be making bacteria food, but instead it injected in our BT gene, and then all I have to do is take this cell and grow it until it becomes an entire plant, and ta-da, I just added an external gene into a plant cell. I transfected the plant. And so this is called the Agrobacterium method because we use Agrobacterium tumefaciens. So basically, in summary, we put our gene on a plasmid, put the plasmid in the bacteria, then we put the bacteria on the plant cell, and then the bacteria uses like a needle to inject the plasmid piece into the cell, and now that cell has our gene, and it will pass the gene on to all of its kids. And we just made a plant cell that we just made a plant that has our new gene and our new trait, hence BT corn or something like that. That's how the Agrobacterium method works. The other method that we use on plants is called the gene gun, which sounds extremely epic, and it is. It's really, really cool. It's amazing that it works, and I can't fathom how somebody figured this out. I can just imagine somebody twiddling their thumbs and they're like, let's try this, and suddenly it worked and they got their PhD and they're like, oh wow. So let me show you how this works. Okay, so the bacteria, sorry, my mind is still back on Agrobacterium here. Okay, here's how this works. We take our little gene of interest here. I'll draw it over here in the corner. Take our gene of interest, and we take a lot of copies of this gene of interest. So we've done PCR on it. There's a bunch of these copies, and we stick them on gold dust. That's right, it's gold dust. They can also use another metal called tungsten, but they commonly use gold. And the reason we use gold is because gold is not harmful to cells. A lot of metals like lead or iron or a bunch of those other ones can be harmful to cells. They're toxic, but gold isn't harmful to cells. And so that way we can use it, but it's not going to hurt the cell in this case. So that's why we use gold. So basically we put the DNA and the gold in this bag or whatnot, and you can shake it up and get the DNA to stick to the gold. So now that the DNA is stuck to the gold, it's going to stay there. Then we take this gold and we put it on the tip of a bullet, like a gun bullet. So we'll take this little bullet here. Okay, here's our bullet. We take our bullet, and our bullet will have like a little divot on the inside, a little pocket, and we'll take these gold particles over here and we'll stick them onto the tip of this bullet. Oops, here we go. We'll stick them onto the tip of the bullet. We'll just pile them all in there. So they're basically sitting in this pouch stuck to the tip of the bullet. And there's just a ton of these DNA-coated gold particles on the tip of the bullet. Then what we do is we take this bullet and we load it in a gun, a legit gun. Sometimes they use black powder. Most of the times it's an air rifle, simply because that's nicer. Basically, think an airsoft gun. So we've got, here's a little gun right here. So it's basically an expensive airsoft gun. Okay, yeah, here's our gun. And then we take the bullet that has our gold on the tip of it, and we load it in the gun. Now the bullet's in the gun, and then we weld a washer to the front of it. So like just your standard metal washer that you can buy at your local handyman store. I'm sure the real things are far fancier, but this is the idea. You take a washer and we weld it onto the front of the gun. So what that does, notice a washer, it has a little hole. It has a little hole in the middle of it right here. So what that does is it essentially narrows the width of the barrel here at the tip. So much so that it will prevent the bullet from escaping the gun. Does that make sense? It's not a complete plug. There's still a little hole in the middle, but it's going to make the barrel at the end too skinny to let the bullet get out. Then what we do is we take a leaf, just like a plant leaf for whatever plant I want to try and change. Over here, we'll take a plant leaf and stick it down on maybe like a piece of metal or something. Then we point the gun at it and pull the trigger. And I just think this would be like the coolest thing to try. You just like point this gun at the leaf, pull the trigger. Now the bullet, when you pull the trigger, it gets blasted down the barrel and gets stuck at the washer. So it flies down there and gets stuck, but the gold particles at the tip are small enough to fit through the little hole in my washer. So they keep right on going at the speed of a bullet. So the bullet was kind of there to help accelerate it. These gold particles come shooting out of here, and they like spray out. This is essentially like a shotgun blast. So you've got like gold particles just blasting all over the place. The whole room's glittering. Everything's going crazy. And basically, as you can imagine, with a shotgun blast, you basically obliterate the leaf. So this thing is absolutely destroyed. The middle of the leaf gets absolutely destroyed. But some of these gold particles here on the outside are going to, rather than annihilating the cell, they're going to punch right through the cell wall, that big armored cell wall. That's why we had to shoot it really hard. They punch through the cell wall and fly into the nucleus of the cell and get stuck there. And the gold particle, again, why we picked gold, it's because it's not going to harm the cell. The gold particle had the DNA that we stuck onto it attached. So when that thing gets blasted into the nucleus of the cell, it drops off the gene in the nucleus of the cell, and now all of a sudden that gene is now in that plant cell. And then we collect the individual cells that survived that got the DNA in the nucleus and grow them. And plants have this really cool ability. A, they're really resilient, so you can shoot them with a gun and they'll come back. And B, they have a lot of stem cells, which means these are cells that are able, you can get one cell and have it grow into a whole new plant. So we basically shoot it with this, and then one of these cells can then grow into a whole new plant, and now that whole plant has this gene of interest in it. Every cell in that plant has the gene of interest, and now I just changed this plant into a full-blown, into a plant that has my genetic, or my added gene, so we turn it into a GMO. That's how the gene gun works. It's legit a gene gun. We use a gun to shoot the DNA loaded on gold particles into the plant cell to change the plant cell. Now, this is kind of a more outdated method. They don't do this very often anymore because the gene guns surprisingly are expensive, and the gold is not surprisingly expensive, and it's very inefficient. Basically, you have to cross your fingers and hope that in the entire leaf, one of the gold particles is going to stop in the nucleus and stay there. And the odds of that happening are very low. And so you might get, if you're lucky, you'll probably get 1 or 2 cells in the whole leaf that will actually work, because sometimes it'll go into the cell and not go into the nucleus, or it'll pass up clean through the cell, or something like that. And so that's the downside to this method is it's not very efficient. You have to pay a lot of money to get very little results. So this one was one of the first ones they tried with plants, and now it's kind of becoming more outdated, and we're using other methods instead. And the Agrobacterium one is definitely the more preferred method now, because it's a lot more efficient. You can get a lot of cells changed in a little bit of time, and bacteria aren't very expensive at all. But it's good to know how the gene gun works for this class as well. So that's how the gene gun works. Let's head back to the PowerPoint, and we'll look at the 3 methods of how to change an animal cell. Because animal cells are interesting. They are not as durable as plants. So we can't use the same methods on them as we can plants, because the Agrobacterium, it only works on plant cells. It's designed by nature to only work on plants, and certain plants at that. And the gene gun, animal cells are a little bit more delicate than plant cells. I mean, if you try to shotgun blast someone in the head, at best they'll have a sparkly forehead, and at worst they'll be dead. Animal cells are very, very fragile, and they don't handle being shot with a gun very well. So they have to have other methods that we use on animal cells that don't work on plants, and vice versa. So the 3 methods over here for animals are electroporation, microinjection, and transduction, or viral transduction is the other name for it. And I'll go into each of these ones in detail as well. Let's start with electroporation. Okay, this is how electroporation works. We've got an animal cell, and we stick it in some water. It's got the cell, it's got a nucleus here. Oh, that's a terrible nucleus. We stick this cell in water, so we've just got water floating around in here. And then we also stick our little gene of interest the gene that we're trying to stick into this cell in the water as well. So we've done PCR on it. We've got lots of copies of it. We dump it in the water. So there's a bunch of it, and we got a bunch of these animal cells in here. Then we electrocute them. As the name suggests, there is electricity involved. We run an electrical current through the water. Essentially, we electrocute these cells. Now, humans don't stand up well to being electrocuted, but apparently one cell can. So if you electrocute a cell, it turns out that the electricity causes these little holes to form in the cell membrane, just all over the cell membrane. These holes are called pores, hence the name electroporation. We had we use electricity pour to create pores in the cell wall, in the cell membrane, I mean. So we've got electricity going on here. There's electricity going on. Electricity. Oh, that's terrible. Okay, it's a good thing I'm not an art major. We've got electricity going on around here. That one's a little better. Okay, lots of electricity. It's making these holes, these pores appear in the cell wall, and it's doing it to the nucleus as well here. Okay, so we've got these pores happening, and the other thing that's interesting about this, and I only recently learned this, you guys don't need to know this part, but I find it interesting. Electricity, we learned, is charged. There's a current, right? So you've got a positive end and a negative end, just like on a battery out here in the water. So the electricity is flowing in a certain direction. And inside the cell, it actually causes the cell to do the same thing. So on one side of the cell, the outside of the cell is going to be positive. We'll make this side positive. And the inside of the cell, so on the other side of the membrane, it's going to be negative. And then on this side of the cell, it's the reverse. You got the positive on this side and the negative on this side. And that's happening all over around this thing. You've got half of the cell that's negative on the inside and half of the cell that's positive on the inside. And then it turns out that DNA, we learned back, clear back in week 2 when we were learning the structure of DNA, that it's got phosphate groups on the backbone, and phosphates are negative. So DNA here is negatively charged. And as you remember, negatives and positives, so negatives and positives are attracted to each other, and two negatives are repelled. So this DNA gets repelled away from that negative end, and it comes around to this side over here, and it gets attracted to that positive end outside the cell right here. So it gets drawn towards the cell at the positive end, and then it gets, it gets drawn towards the positive here. So we pull it inside the cell. Sorry, I have to think about this one here. It's going to get pulled inside the cell with the charge. We've got the positive charge here, and then it gets to that positive charge there, and then it feels this positive charge over here, and so it gets then dragged through the hole, being attracted to that positive charge there. And now we got our DNA inside the cell. Sorry about that. I hope that was easy to follow. Thankfully, you don't need to understand this piece. So we got our little gene of interest right here. Now it's inside the cell. Now all we have to do is turn off the electricity. So we take the electricity away. The electricity goes away. That means the charges go away. The charges go away. And when the charges go away, that means the electricity is not forming holes in the cell membrane anymore. So the cell membrane fixes itself. We come in here and we fix all of these holes. And now the DNA that we dragged inside the cell is trapped inside the cell. And hopefully we'll get it to also go inside the nucleus too. Because essentially it's just like a little, in theory, it's like a cell within a cell. You've got a little membrane there. So you just drag it through there and then drag it inside the nucleus until you got it inside the nucleus. So the idea behind this one, again, you don't need to remember all those particulars. I just included them because I find it fascinating. And I'm sorry if I confused you there. But the idea behind electroporation is you put the DNA in with the cell in water, you electrocute it, it forms holes in the membrane, the DNA gets dragged inside, and then you take the electricity away, the holes fix themselves, and now you just got the DNA inside the cell. That's how electroporation works. And again, we only do this one on animal cells because the plant cells, they've got their cell wall, so the DNA wouldn't be able to get in. Let's go to the next method here. The next method, like the name suggests, microinjection is one of the easiest ones for students to get their minds around. Everybody's gone and gotten a shot at the doctor. That's exactly what we're doing here. We're just doing it on a really small level, hence the name microinjection. The bacteria, or the cell, the animal cell that we're trying to put the gene in, is basically just getting a shot. Here's how it works. We've got a cell here with its nucleus, and then we've got a needle over here, a really, really skinny needle with just like your standard syringe up here at the top. And in the syringe, it's got our gene of interest. So our DNA that we want to add is inside the syringe, inside the needle. And then they basically use this vacuum. If you ever get a chance to watch one of these, it's really cool. They use this tiny vacuum to hold on to the cell. So it comes in here and it's basically just a little tube. And you use the vacuum to hold the cell still so that you can work with it. So there's using the suction of the vacuum to hold the cell still. And then we take the needle. So this is all under a microscope because cells are really small. They take the needle and poke it into the nucleus of the cell. And when you poke it into the nucleus of the cell, then they inject the DNA. So it's got a syringe on it. They inject the DNA into the nucleus of the cell and then pull the needle out. And then that means you've got the DNA inside the nucleus of the cell. The gene that you wanted to add, now it's inside the nucleus of the cell, and ta-da, we got it too. We got the gene inside the cell. You pull the needle out, the cell's perfectly fine. You implant it in a mother, and it grows into a full organism. The downsides to microinjection and to electroporation is you have to do it on only one cell. So it's got to be like a zygote, a fertilized egg. That you're doing this to, because you can't just go and poke every single cell in a human's body as they're an adult. That would A, take forever, B, extremely painful, and it wouldn't be effective. You got so many cells and you're very thick, like it's just not feasible. And if you electrocute a human, it kills them. So we've got to use other methods, or we have to develop a method. This method works great if you're working on a zygote, one cell that you then implant in the mother and it grows into a whole organism that has our new gene in every cell. That's great. But we have to have another method if we're going to change an animal that is already alive, has already been born, because these first two methods won't work. Hence why we go to our third method here. The last method is called viral transduction, or simply transduction is probably how you would most commonly see it. And transduction is, again, us commandeering a natural process. So remember, these are, it's kind of savage vocabulary-wise because they all sound the same. It's not translation. Translation was when we read the mRNA and turned it into a protein. It's not transcription. That was when we read the DNA and turned it into RNA. It's not transformation, because transformation was getting the plasmid from outside the bacteria to inside the bacteria. This is transduction, like a duck. It's transduction, and the definition of transduction is using a virus instead of a plasmid. We're using a virus to deliver the gene of interest into the cell. And this has a couple of perks here. Basically, let me show you how this works. Here's basically how it works. We've got our cell here. Now, viruses, are technically, by the definition, not alive. Most people don't know that, but by scientific definition, a virus is not alive, because it doesn't have the ability to make, or it breaks a couple of the rules, actually. It doesn't have the ability to make its own babies without somebody else's help, and it doesn't have the ability to run metabolism. It doesn't burn sugar or anything like that. So technically it's not alive, which is just a fun fact. You don't need to know that, but let's pretend that this is my virus. Viruses are actually much, much smaller than this relative to the cell, but oops. But here's my virus. Viruses work by finding a protein, and the protein varies between viruses. Let's see here. Go back to here. Yeah. It finds a protein that pokes on the outside of the cell. It just sticks out like an antenna. And there are lots of proteins that stick on the outside like an antenna. For example, and each virus looks for a different protein on the surface of the cell. So for example, the recent COVID-19 pandemic, the virus SARS-CoV-2 binds to a protein on the surface of lung cells called ACE2. That's a receptor. And so this virus comes in here and it sticks to the protein that it looks for. It sticks to the outside, and these proteins, they're receptors usually. They're designed like mailboxes, and their job is to wait until some molecule that they're designed to look for passes by, and they grab onto it and they say, aha, and they pass a message to the inside of the cell. And sometimes they cause the cell to bring in the receptor and whatever's attached to it to just bring the whole thing inside. And usually it's a good thing, because whatever you're bringing inside, it could be like sugar. Like, let's say sugar was supposed to bind to this stick, and then the cell brings it inside so that it can burn it. Well, in this case, the virus is basically doing a Trojan horse scenario. The virus tricks the cell by looking to the protein. It looks like whatever it was supposed to be binding to. And so it sticks to this protein and convinces the protein it's whatever message it was supposed to deliver. And so then the cell brings the virus and the protein inside. And then the virus is like, boom, surprise, it's me, and breaks open and drops off the DNA that it's carrying. So the virus breaks open. This is how viruses work naturally. The virus breaks open and releases its DNA into the cell. And then it uses the cell to make more viruses. So it uses the cell's ability to transcribe and translate proteins to make more viruses. And it uses the cell to divide its DNA and create more DNA, so that now the cell makes lots and lots of virus babies. And then they escape the cell and go infect other cells. That's how a virus works. Our idea is to commandeer this process by using a virus. What if we can make a virus that, rather than having bad virus DNA on the inside, has whatever gene I want to drop off? Whether that be the factor VIII gene to help cure someone from hemophilia or something like that. We take out all the bad virus genes that get you sick and make baby viruses, and we instead put in whatever gene we're trying to drop off, and then viruses just spontaneously form. So basically we can put this in there with these virus capsules, and then they get inside the capsule, and today you just made these perfectly good viruses that can't make more viruses. It's really trippy, actually. It's fun though. So you can make these viruses that have your gene of interest, and the virus is designed to get inside this cell and drop off the DNA inside of it. So this virus, then I can put it inside an animal, and it will go hunt down its usual receptor, just like it would if it was getting you sick. It would go hunt down the receptor and stick to it. It would trick the receptor and have the cell bring it inside, and then it'll break over and be like, pop, I've got my DNA, ha 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 ha. And it turns out, since we tricked the virus, rather than dropping off the virus DNA, it drops off our gene of interest. And ta-da, I just got the DNA inside the cell, and it's not gonna make new virus particles, so I didn't get you sick. And now that cell has that gene in it, and I just transfected the cell. I just changed the cell turns the cell into a genetically modified organism. Now, there are lots of different kinds of viruses, and all of them have upsides and downsides. There's 3 specific categories of virus that we ask you guys to know about that we use as viral vectors for various reasons. So let me jump back over to the PowerPoint, and we'll go over each of those different categories of viruses. Excuse me. We'll go over each of those categories of viruses and what the upsides and downsides to each category are, so that you can know how those work. Okay, so here's the table. They're the 3 groups of viruses we ask you to know about. So these are animal viruses. The 3 groups of viruses we ask you to know about are the lentiviruses, the adenoviruses, and the adeno-associated viruses. Sometimes that's abbreviated AAV. So if you see AAV, that's the adeno-associated viruses. So let's go through each one of these in detail. Now, all of them work on the same principle. Now we just have to go and elaborate on the details of each one. So let's look at the lentiviruses first. An example of a lentivirus is HIV. Now, lentiviruses, you might recognize them more by the name retrovirus if you've read that in the reading or heard it. Lentiviruses are a subclass of retrovirus. So the lentiviruses are retroviruses. This is just the more specific name for these particular types. HIV is a lentivirus. These are fascinating. They actually have a protein, they actually carry a protein that breaks the rules that scientists thought how this worked. In this class, we taught you central dogma, clear back at the beginning, where the flow of genetic information goes from DNA to RNA to protein, right? Well, these cells carry these viruses, I mean, they're not cells, they're viruses. They carry a protein called reverse transcriptase. Let me actually pop over and show you a picture. Okay, so this is a picture. These are HIV viruses. So this is a really powerful microscope. It's called a transmission electron micrograph. So this is a photograph of real virions. Each of these circles with the dark triangle in the middle, that's an HIV particle. It stands for, HIV stands for human immunodeficiency virus. It causes the human disease AIDS. So that's what they look like, and they carry this fantastic enzyme called reverse transcriptase. And this is what it looks like. You don't need to know that. I just find it fascinating. That's the structure of how this thing looks. And what it does is, as the name suggests, reverse transcriptase, it does transcription backwards. So unlike your normal cells, or excuse me, cellular processes, where you go from DNA, you go DNA to RNA to protein, this enzyme, let's go here, DNA to RNA to protein, this reverse transcriptase has the ability to read an RNA and make DNA out of it. So it's actually able to make double-stranded DNA by reading an RNA template. And this just blew scientists' minds, and we actually use this for a lot of really cool things. If you want to learn more about how the scientists use reverse transcriptase in research, it's really cool. But basically, these HIV viruses, they have an RNA genome. All their genes they hold them inside the virus as RNA. So the HIV pops into the cell and drops off its RNA. But it doesn't just stop there. It drops off reverse transcriptase. And the reverse transcriptase takes the viral RNA. So let me put that in red. So here's the HIV RNA. And, oh boy, that was exciting. And turns it into HIV DNA. This way. So now we've got double-stranded HIV DNA, which is the same thing as the RNA. It's got the gene and everything. We just went backwards and made DNA instead. And then it goes one step further. And it takes that DNA inside the nucleus of the cell, and it has a restriction enzyme called integrase, spelled like this. Integrase. It uses the integrase enzyme, and integrase, as you remember, it's a restriction enzyme. It cuts the DNA, so it cuts your chromosome, cuts it open, glues, and then glues in the DNA, the HIV DNA, into your chromosome. What that means is the cell, it's now a permanent part of the cell. So the cells, your chromosomes are making the virus now. And there's no way to get it out now. It's a permanent part of the cell's chromosome. And when that cell divides, it divides the viral DNA too. And so it copies the viral DNA, passes it on to all of its kids. So if one cell gets infected, all of the cells that get made off of that cell also are infected. Isn't that clever? So in effect, if you get infected with HIV, this is why HIV is a lifelong infection. Because the HIV virus gets inside your bloodstream and goes after your immune cells, specifically helper T cells. The protein that it sticks to is only found on the surface of immune cells called helper T cells. And it gets inside them and it glues the instructions on how to build an HIV virus into the T cells' chromosomes. And the only way for us to get, or to cure you of HIV is to kill you, because the cells, because the cells themselves, it's the chromosomes that are making the virus. So the only way to do that, or to get rid of the virus, is to kill you. And we don't like doing that. So most treatments now for HIV are trying to slow the virus down, prevent it from binding to new cells, prevent it from escaping the cells that's already infected, things like that, trying to prevent it from spreading to other cells. But we can't cure you of HIV until we can figure out how to get that HIV DNA back out of your helper T cells. So that's why HIV is that's why HIV and AIDS is such a big deal, because it's a permanent disease, because it glues its DNA in. Now, with that bit of background here, let's go back to our table. This will help you hopefully help it make more sense now that you've got the background. The way that we use the HIV is we take out all of the bad virus genes, and we glue in whatever gene we're trying to insert into the animal. And then we infect them with the lentivirus, HIV or something like it. And it goes into them and it binds to their cells, gets taken on the inside, and then it uses reverse transcriptase to read the RNA copy that we gave to the HIV of our gene of interest, make a DNA copy, and then use integrase to glue it into the host chromosome. Now, with HIV naturally, that's a bad thing, but with our GMO version, it just permanently glued the gene in. So for example, if we had a version of a lentivirus that could infect liver cells, then I could take out the bad HIV genes and put in a healthy factor VIII gene, and then inject a person with hemophilia with this virus, this modified HIV virus. And hemophilia, if you remember from a couple of weeks ago, that's caused because they can't make a healthy factor VIII. They can't make factor VIII, and so they can't clot their blood. And the liver is the organ of the body that normally is able to infect, or is normally building factor VIII. So if we got a lentivirus that could infect the liver, we put the factor VIII gene on the inside and take out all the HIV genes, and then it goes and binds, gets carried inside the liver cells, and then it permanently glues in the good factor VIII gene into that person's chromosome, and now they're permanently cured of hemophilia. Their cells now have a working version, all the cells in their liver now have a working version of factor VIII protein, and I just cured them permanently of hemophilia for the rest of their life, because when the cells divide, they'll pass the copy on to their kids because it's part of the chromosome, and ta-da, we just permanently cured that individual of hemophilia. And it's extremely good at it too. That's what the high efficiency is. Like if you stick it in there, it's pretty dang good at getting that DNA inside your chromosome. So that's fantastic. We like that. There are downsides though. Like everything good, there are downsides. One downside is HIV, it's got RNA on the inside of it. And reverse transcriptase is actually atrocious at, it's not very efficient and accurate. So it makes a mistake. Well, it makes a mistake like every 5,000 bases. So there, it's got an extremely high mutation rate. Normal proteins that are making DNA make a mistake about once every 1,000,000 bases. So this reverse transcriptase helps HIV mutate really, really fast. And that's an advantage for the HIV virus naturally, because it helps it keep ahead of your immune system, because the immune system is constantly having to try and figure out how to beat the next version of the virus. But that's bad, because if it mutates so quickly, I could accidentally have the gene, or the factor VIII gene, mutate between the time that I put it in the virus and inject it with you. And now all of a sudden the treatment won't work anymore because I just injected you with a mutated form of the factor VIII gene, because the virus wasn't stable enough to keep it steady long enough to inject you with it and get it into the chromosome. That's one of the downsides to these lentiviruses, is they have a really high mutation rate. So you're basically crossing your fingers that the gene will last long enough to get it turned into DNA and glued into the chromosome, because DNA is a lot more stable. It's just RNA and this reverse transcriptase that cause our problems. Another big problem that makes us very hesitant to use these lentiviruses is it inserts the DNA in random places. So in week 10, you learned that restriction enzymes have to glue or have to cut at a specific sequence. Well, integrase is different. Integrase is this all for one and one for all restriction enzyme that cuts wherever it darn well pleases. So we actually can't control where integrase cuts. It just cuts wherever's convenient for it and inserts the DNA in there. Now, if it cuts in junk DNA, great. It will cut the junk DNA, which doesn't harm the cell at all. It'll glue in our factor VIII gene, close it back in together. Ta-da, we just cured you of hemophilia. But what if it accidentally cuts in the middle of an important gene? We learned in week 5 that if you break a gene, you break the protein. It's not going to work anymore. You can't transcribe a gene that's ripped in half. So if we cut the sentence in half and insert a sentence in the middle, you can't read the original sentence anymore. The cell can't read it anymore. So if, for example, that it cuts in the middle of a gene that's responsible for preventing the cell from dividing too often, then I could cure you of hemophilia and give you cancer at the same time. We don't like those odds. It wouldn't happen every time, but we don't like the odds of potentially giving you cancer while trying to cure you of a genetic disease, or causing another genetic disease in the process of curing you of a genetic disease. That's a little bit of a downside there that we like to avoid. The other disadvantage is HIV is recognized by the immune system. And so the immune system calls out the army and comes and tries to kill the thing. And if it does too good of a job, it will kill all of the virus particles I inject into you before they can actually infect your cells. And so your immune system actually wipes out my treatment before it helps you. That's a bit of a downside, especially if you get too strong of an immune response, it can actually kill you. So if we're not careful, you could react extremely strongly to the virus and throw you into septic shock and kill you. So we don't like things that trigger your immune system very powerfully. A, because it could destroy the thing that I put in you to try and help you, and B, because it could kill you in the process. So we try to avoid that. So those are the upsides and downsides to lentiviruses. And an example of a lentivirus is HIV. The next category are the adenoviruses. If you've had any medical experience, you've probably heard of the tonsils. The tonsils are at the back of your throat, and some people have to get tonsillectomies to have them removed because they get constant strep throat infections. Well, at the back of your nose, there's tonsil-like organs called adenoids. And the adenoids sometimes have to be removed with the tonsils as well. But adeno, in the name adenovirus, refers to the nose. With the adenoids, the nose. And so these viruses remember, cells target viruses target very specific cells. And so the cells that these adenoviruses go for are respiratory cells. So nose, chest, or nose, lungs, throat, things like that. And so an example of the adenoviruses are the common cold. There are lots of viruses that cause the common cold, but a bunch of them are adenoviruses. And so these are much more tame than the HIV ones, because as you notice, getting a common cold is not a lifetime event. You know, it lasts about a week and a half, and then your immune system takes care of it, and it's gone. That's because adenoviruses, they don't glue their DNA into your chromosome. So there's no chance of giving you, so there's no permanent damage. There's no chance of giving you any cancer or anything like that. I take my gene, I put it inside the adenovirus, I infect you with the adenovirus, it goes, binds to your respiratory cells, goes on the inside, drops off our gene of interest, but it doesn't glue it into the chromosome, so I don't have to worry about giving you cancer in the process like I do with the lentiviruses. The downside is, because I'm not permanently gluing it into the chromosome, it doesn't last forever. Remember, the cells only replicate the DNA that's in their chromosomes, and so it's not going to pass it on to any of, once the cell divides, it's not going to give the viral DNA that I dropped off or the modified gene that I dropped off in that cell to the new cell. And eventually it'll just destroy that piece of DNA that I dropped off. So basically, using an adenovirus, I could safely cure you of hemophilia for a week. Assuming I had an adenovirus that could infect the liver, of course. Which we can alter them to probably do something like that. But I could cure you for about as long as the common cold lasts, and then it would be gone, and you'd have hemophilia again. So that's the downside to not gluing it into the chromosomes, is yes, it's safe, but the changes are temporary because I'm not gluing it into the chromosome, so eventually it wears off after a week or two. And again, adenoviruses are recognized by the immune system, and we don't like that because A, you could destroy the viral vector before it gets there, and B, it could have potentially harmful effects if the immune system gets out of control. So that's the difference between lentiviruses and adenoviruses. The other one with adenoviruses, though, is they have DNA as their, they have double-stranded DNA usually as their genetic code, and DNA is far more stable than RNA, so they have a low mutation rate. I didn't add that to my table here. I'll add it to my table. Okay, there we go. Low mutation rate, it's on my table now, there we go. So they have a very low mutation rate. They don't change very often, so I'm pretty confident that the gene I put in there is going to last long enough to be safely incorporated into the cell. I just can't get it to stay permanently there. That's the downside. So now we'll go to the third category, the AAVs, the Adeno-Associated viruses. Now these ones, I don't have an example for you. And part of that is because they're not very well known. They're actually, we recently discovered them. So we're still learning about them. These viruses are extremely tiny, even for viruses. And they're really trippy because they're like a virus for viruses. They infect, or they basically are parasites for viruses. So Adeno-Associated, the name, means that these viruses only work if they're with an adenovirus. Let me show you what I mean. Okay, hopefully I can help this make a little more sense here. We've got a cell. Let's pretend this is like in your lungs or whatnot. We've got an adenovirus, so not an adeno-associated virus. This is an adenovirus. So we'll say this little brick here. Let me change colors here so that we'll make this the adenovirus. Okay, we've got an adenovirus, and normally an adenovirus can go inside your cell and get you sick. And it uses the adenovirus uses your cell's machinery to make more baby adenoviruses, and then they get released to go get other cells sick. Well, these adeno-associated viruses, so let's put in, I'll make these ones a little green circle here. Make them green. Okay, there's our adeno-associated virus. These adeno-associated viruses can also pop into cells, just like a normal virus, but they can't make new baby viruses by themselves. Even with the cell's help, just an adeno-associated virus, if it infects your cell, it can't make baby viruses. In fact, it has to wait until an adenovirus also infects the cell at the same time. And with the adenovirus's help and the cell's help, now we can make baby adeno-associated viruses. But that's why we call them adeno-associated viruses, because they have to have an adenovirus that infects the cell at the same time as they do in order to work. So they're associated with adenoviruses. So these adeno-associated viruses are fantastic. They're really, really cool as viral vectors. Let's go back to the table to show you how that works. So basically, just to go back over this in a visual way here, let's make a table. If you have just the adenovirus, you get sick. If you have just the AAV, you don't get sick, because the virus can't get you sick. If you have just if you have both the adenovirus and the adeno-associated virus, then you get slightly more sick, I think. But really, this adeno-associated virus doesn't really seem to have too much of an effect. If anything, it gets you slightly more sick, but at the least, you just get your standard cold. As far as they know, these adeno-associated viruses don't get you sick. They have yet to find an example where an adeno-associated virus gets you sick. So it's extremely safe. Let's pop back over to the table here. It's extremely safe because it can't get you sick. So you don't get sick. The immune system actually, in most cases, there are occasionally people that recognize it, but the immune system ignores them. So you don't usually get an immune response, and if you do, it's not very powerful. So the viruses, the little adeno-associated viruses that I put my gene in and infected you with, they don't get, get recognized by the immune system, so they won't be destroyed, and they won't risk harming you with your own immune system like the other two can. And here's the fun part about these. They actually incorporate their DNA into the chromosome just like lentiviruses do. So they make a permanent change by gluing their DNA in. But unlike the lentiviruses where integrase glued them wherever it darn well pleased and we risk giving you cancer, adeno-associated viruses only glue in their DNA in a specific spot. They actually don't use a restriction enzyme. They use a different method. But they are able to, with a high degree of efficiency and accuracy, they only glue their DNA into one spot on chromosome 19 that happens to be in the junk DNA. So these viruses don't cause cancer, they don't get you sick, they don't alert the immune system. It's like, whoa, what's the downside? Like, this is basically the best of both worlds. You get all the advantages of the lentiviruses with all the advantages of the adenoviruses. It's like a win-win situation. The major downsides to the adeno-associated viruses are they have a very low carrying capacity, meaning these things are tiny. They're small even for viruses. Like viruses, normal viruses are colossal compared to these things. They're just really, really small. And if you imagine, a virus is basically a protein bubble filled with, it's like a box basically filled with DNA. And the bigger the box, the more you can put in there. And so if you've got a gigantic box, you can put a lot of stuff in there. And if you've got just a little tiny box, you can only fit a few things in there. These adeno-associated viruses are just physically tiny. And because of that, there's very little room on the inside of the virus. And humans' genes are really big usually. Human genes are very, very long. And in fact, genes, as a general rule, most genes for animals and plants are traditionally very big on average. And so most of the time you won't be able to fit them inside the adeno-associated virus. So you can only use this for limited applications because the adeno-associated virus is simply too small to be useful for anything that's big. The other downside is these adeno-associated viruses. Remember, the only way to get baby adeno-associated viruses, because remember, we have to have the adeno-associated virus to put the gene in the first place. So we have to have an adeno-associated virus to modify in order to infect the person with it. But in order to get the adeno-associated virus, remember, it can only make baby adeno-associated viruses if it also has the adenovirus infecting it. If we go back to our picture, notice remember, the adeno-associated virus can only make baby adeno-associated viruses if there is an adenovirus also there. So the only way to get adeno-associated viruses is to take some cells, put them on a petri dish, try to keep them alive, because human cells don't stay alive very well on a petri dish like a bacteria does. So you have to try and keep them alive, and then you have to infect them with the adeno-associated virus and with an adenovirus at the same time, and then have the cells spit out adeno-associated viruses and adenoviruses, and then you have to sort out what's adenovirus and what's adeno-associated virus. Then you have to take the adenoviruses apart, put the gene in, and then glue them back together again, and it's just a nightmare. So that's the other downside to these adeno-associated viruses, is they're just a hassle because they're much more difficult to make than the other ones, because they rely on other viruses to make them. And so it's just this big, huge hassle. It's not impossible, it's just a hassle and not very efficient. And then again, for all of these, so those are the three main categories of viral vectors that we use with some examples, the upsides and the downsides of each one. Those are all good to know. You should know those for the exam. One downside to all viruses as vectors, the upside is you can use them in full-grown after-birth creatures, because they infect. You can inject them into them and they infect a bunch of different cells, and so you're able to change more than one cell at a time, like the other two methods, the electroporation and the microinjection. But the downside is, remember, those viruses are extremely cell-specific. So I can't change the gene in every cell of your body. I can only change it in like one organ or one tissue, for example. Like I could add the gene in your lungs, but not in your heart. I would have to have a different virus to go do it for your heart, etc. So that's a downside to all viruses as a viral vector, is simply they're extremely specific. And so they only attack the cells that they're designed to attack, and they don't attack any other cell. And so it's difficult making widespread change outside of one tissue type or one cell type or one organ, because these viruses simply ignore all the rest of the cells. So sorry, that was a lot of information. This was a really long video. Feel free to, you're welcome to go back over it and pause it, watch different segments and stuff. But definitely these are good things to know. Understand how each of these methods work. And for your presentations this week, for the 6 genetically modified organisms, they ask you guys to identify which of the methods, which of these 5, go back over here, which of the 5, too fast, which of these 5 methods of eukaryotic transfection were used to make that particular GMO. And now you know the background behind how each of those 5 work. And you should definitely be able to recognize what it is with the name and how it works, kind of thing. So email me at Dallin, dallin.stokes17@gmail.com if you have any questions. And I'm glad to help you out. Feel free to ask your professors, ask your TAs. We're here to help you. So hopefully this video helps clarify a bit of the reading, and have a wonderful week.